what is a virus? Viruses, they are not cells. They cannot replicate themselves independently. They cannot synthesize their own energy or their own proteins, and they are too small to be seen in the light microscope. So here we say that they are not cells, but actually what are they? Now they are made up of, there's an internal core which can either be due to DNA or RNA, the capsid, which is the protein covering or the coat over the RNA or DNA, and then there's an envelope, which is the outer lipoprotein membrane, which is external to the coat. The viruses, they do not have a nucleus, cytoplasm, mitochondria or ribosomes. They cannot replicate independently, therefore they are known as obligate intracellular parasites. Now, they are not undergoing binary fission or mitosis. Where do we find binary fission? In bacteria, and the mitosis in the human cells. But they produce hundreds of progeny viruses. Now this is the structure of a virus, and we can see here this is the nuclear core, which can either be DNA or an RNA. So this is a nuclear core, and this can either be a DNA or RNA, but can never be both. Now then, the capsid, which is the protein covering or coat. So this is the capsid here, blue in color, geometrical shape here, and this covers this nuclear core. And then we have the envelope, which is the outer lipoprotein membrane, and here you can see over the this capsid is an envelope. Now envelope may have spikes here for the attachment to the host, but on this side you can see that the envelope is not present. So therefore we say that there are two types of viruses. Either they can be enveloped with regards to their covering, and they can be non-enveloped or naked nucleocapsid viruses. So here you can see an envelope virus, and here you cannot see an envelope. Now how do we compare viruses with cells? Don't you think it's better to understand what is the difference between cells and the viruses? Now in viruses, the type of nucleic acid, they can either be RNA or DNA, but never both. But in cells, we find both DNA and RNA inside it. Now proteins are very few in the viruses, whereas there are many proteins in the cells. The lipoprotein membrane, the envelope which is present in some viruses, it is made up of lipoprotein membrane, whereas in cells, entire cell membrane is present in all cells, and it is made up of lipoprotein membrane. Ribosomes and mitochondria are absent in viruses, whereas they are present in the cells. Now mitochondria are present in the eukaryotic cells, which are the human cells, but not in the prokaryotic cells, which are the bacterial cells. So you have to keep this in mind. The enzymes, which are very few in the viruses, whereas there are many enzymes in the cells. And multiplication by binary fission or mitosis. The viruses, they do not undergo binary fission or mitosis, but the cells, the human cell undergo mitosis, whereas the bacterial cells undergo binary fission. Now the size of the viruses can be from 20 nanometers to 300 nanometers. The shapes can be spheres, rods, bullets or bricks. So here you can see a bullet shaped virus, which is the rabies virus. Here you can see a rod shaped virus, which is an Ebola virus, and here you can see a sphere shaped virus, which is a coronavirus. Now viral nucleic acids, they are also known as genomes. They can be single or double stranded DNAs, or they can be single or double-stranded RNA. The nucleic acid can be linear or circular. The DNA is always a single molecule, whereas the RNA can exist as a single molecule or it can exist in several pieces. So in influenza virus or rotavirus, it is composed of segmented RNA genome. All viruses are haploid except the retrovirus, which has 2 copies of the RNA genome, so therefore they are known as diploid. Otherwise, all viruses are haploid. Now, viral capsid and symmetry. The nucleic acid, as we have seen, it is surrounded by a capsid. The nucleic acid combined with the capsid is known as the nucleocapsid. So here you can see in the diagram that the nucleic acid combined with the capsid is known as the nucleocapsid. Okay. Now the viral nucleocapsids have 2 forms of symmetry. Either it can be icosahedral or it can be helical. So this geometrical shape here is the icosahedral shape. So this is the helical shaped viruses. All human viruses that have a helical nucleocapsid, they are enclosed by an outer membrane called the envelope. So there are no naked helical viruses. And viruses that have an icosahedral nucleocapsid can either be enveloped or naked. Now this is a filamentous or helical nucleocapsid, and this is a virus which is having an icosahedral shape. Now viral protein functions. The capsid protein protects the genome, DNA or the RNA. So this capsid here, it is protecting the nucleic acid. The surface protein mediates the attachment to host surface membrane and also determines the serotype. So this virus which is having spikes, and nowadays the coronavirus which is a hot issue nowadays, it is having spikes on its surface. So it is important to mediate attachment to host surface, and it also determines the serotypes. Outer viral proteins induce neutralizing antibodies and activate cytotoxic T cells to kill virus-infected cells. Now, what is a viral serotype? It is a subcategory of a virus based on its surface antigens. Measles virus has one serotype, that means only one antigenic determinant on its surface protein. So when you want to get yourself immunized against the measles virus, the vaccine in it can have just one type of antibodies to neutralize this one serotype, and hence then you are protected. But in contrast, the polio virus, it has 3 serotypes. That means there are 3 different antigenic determinants in polio virus. Type 1, which has one kind of antigenic determinant. Type 2, which has another different antigenic determinant. And the type 3 polio virus, which has a different antigenic determinant, which is different from type 1 and 2. So, if a person who wants to be immunized against the polio virus 1, if he is immunized against the polio type 1 virus, he can still get the disease poliomyelitis caused by polio virus type 2 and 3. So, for a person to not have this polio virus disease by any type of serotype, the polio vaccine must contain all 3 serotypes in order to be completely protective. Now, what is viral envelope? It is a lipoprotein membrane. It is a lipid derived from the host cell membrane, and the protein is virus specific. Now, the lipo portion or the lipid portion here, this is coming from the host cell, and it forms the envelope, and the protein on it is virus specific. Like spike-like projection on the surface, they are viral specific, which attach to the host cell receptor during the entry of the virus in the cell. Now the virus envelope is acquired as the virus exits from the cell. in a process called budding. Now, when it wants to invade other human cells, it just comes out of the human cell, taking its cell membrane and converting it into its own envelope. In general, the presence of an envelope confers instability on the virus. So, all the envelope virus, they are unstable virus. Now, what does that mean? What is the difference with regards to envelope or non-envelope viruses? Now, envelope virus, they are more sensitive to heat, drying, detergents, lipid solvents like alcohol than the non-envelope viruses. So, all these envelope virus, they are unstable viruses and they are more sensitive to these agents. Now, viruses which are transmitted through fecal-oral route, they do not have an envelope, so they are environment survivors. For example, hepatitis A, poliovirus, Coxsackie virus, ecovirus, Norwalk virus, and rotavirus. Viruses transmitted by blood or sexually transmitted routes, they are envelope virus, so they are less stable, like HIV, herpes, simplex virus 2, and hepatitis B and C. Now, the respiratory aerosol droplets like influenza virus, measles virus, rubella, respiratory syncytial virus, and varicella zoster virus, they are envelope virus and they are rapidly inactivated. Except for the rhinoviruses, they are naked nucleocapsid virus and can survive in the environment for significant period. Now, the DNA viruses, you can look at the chart in your Levinson. There are double-stranded DNA virus and there are single-stranded DNA virus. The double-stranded DNA virus can be naked or enveloped. The naked are the adenoviruses, papilloma virus, polyoma virus. Enveloped are hepatinovirus, herpes virus, pox virus. And single-stranded DNA, which is naked, is parvovirus. RNA virus, single-stranded naked, we have calicivirus, herpesvirus, picornavirus. Enveloped, single-stranded, coronavirus, which is the hot issue nowadays, flavivirus, retrovirus, togavirus, arenavirus, bunyavirus, filovirus, and so on. Double-stranded RNA, we have the reovirus. Thank you, students.